Okay, welcome to an SJ Brothers video, and I'm going to be talking about, let's see, hope it's still recording, yes it is. I'm going to be talking about, stuff. Yeah, that's what I'm going to talk about, stuff. Well, more specifically, cell bio stuff. And about, basically the methodologies and microscopy techniques that are used. Uh, the techniques first of all. In a separate video, I will show, go into more detail about the types of microscopy, like light fluorescent microscopy and also electron microscopy. But for this video, I'm just going to be focusing on SDS page, peptidases, immunofluorescence, immunoblotting, and immunoprecipitation. So, first of all, let's talk about SDS page. SDS basically stands for sodium didecyl sulfate, and it's an amphipathic or amphiphilic molecule, basically meaning that it has both the hydrophobic and hydrophilic region. More specifically, the sulfate head group is actually the hydrophilic part of the molecule, and the hydrophobic part of the molecule is the long, really long hydrocarbon tail, which can be seen with all those nice dashes, in case you haven't taken organic chemistry. Um, so, uh, the method itself basically relies on the sieve, which is basically the gel. And what happens is that the amount of SDS bound to the protein, which has hydrophobic regions, due to hydrophobic residues actually, but due to like hydrophobic interactions, they aggregate together. And so more SDS will be bound, meaning that there'll be more negative charge on the protein because of the sulfate head group as well. And there'll be more indicating that the length of the protein is actually longer and the molecular mass of the protein is actually more. These are key things that I kind of picked up in my cell bio class. My professor was on about that. Okay, so maybe yours might. Um, but however, the key thing about the SDS page is again the sieve. So due to the fact that they have negative charges, irrespective of how big the molecule, like the protein is, they're going to, or how small it is, it's going to migrate its way, or find its way towards the positive end, which is the anode in this case. So the deciding factor is how well the protein can actually migrate through the gel, and this is smaller proteins will migrate a lot faster than bigger ones. So we tend to see bigger ones at the top and smaller ones at the bottom, basically. Okay, and that's some hints about SDS page. Okay, moving on, we're going to be talking about peptidases, or otherwise known as peptide bond cleavers. And basically, the one that we talked about in my cell bio was tryptic peptides, which is caused by trypsin, which is an enzyme in our stomach, and we use it all the time. Great. Okay. Um, so, coupled with mass spectrometry, like once you get tryptic peptides, which usually trypsin acts like at the C terminus of lysine and arginine like residues. So once you get that, you put your tryptic peptides in the mass spectrometer, and each of them have unique mass to charge ratios. Z shows the charge, and this is now matched up with protein databases and can then be used to identify what type of protein it is, or get a rough estimate of what it is, basically. Okay, that's what I want to say about peptidases. Moving on to immunofluorescence. I'm not really going to go in detail with immunoblotting because I feel like it's said in both immunofluorescence and also in immunoprecipitation. Basically, for immunoblotting, you're transferring the SDS proteins on the SDS gel, or rather the page gel, um, onto a nitrocellulose support. Okay. Uh, Anyway, so immunofluorescence involves primary antibodies, secondary antibodies, and involves fluorescence tags attached to secondary antibodies. Typically, we don't usually use fluorescent tagged primary antibodies because of signal amplification, and also due to the fact that it's kind of more annoying to design a specific type of primary antibody that has a fluorescent tag attached to it that detects a specific antigen. Well, did I say that right? Yeah, the primary antibody that detects a specific antigen, which is a protein, by the way. And with this method, we can also like view specific types of proteins in like under a fluorescent microscope, which I'll go into more detail in another video, provided this goes well. Uh, so, moving, let's see, the drawback to this technique, though, is that it can't really be used to determine like if proteins are associated with one another, and also it does involve killing the cell itself. So it's not very helpful to see how proteins actually move or function within the cell. Just kind of get like a location of where it is. Um, yeah. Okay. So but to counteract the drawback of the association between proteins, we use immunoprecipitation, which basically just involves a test tube. That's in this diagram involves a test tube with a couple of proteins in it. In this case, it's going to be the green and the red, and basically they associate with one another. An antigen is used to detect, or rather, couple to the an antibody is used to couple to the green protein in this case, and that antigen is actually coupled with a bead. So basically, when this test tube is spun in a centrifuge due to weight, the associated proteins coupled with the antibody and the beads are found at the bottom of the test tube. And so we can then run this through an STS page, and we'll find that actually there are two bands. If there are two bands, it indicates there are two types of proteins in this case. So anyway, hopefully that helped, and hopefully I wasn't ranting too long. But yeah, if that's clear to you and if that helped in any way, please let me know. If it didn't, also please let me know, but with caution. Anyway, so yeah, I would be making the second video on microscopes and give a brief overview of that too, if this goes well. Alright, thank you for watching, and stay tuned in for another SGA Brothers lecture.